Welcome to today's webinar, New Discoveries in Brain Technology, the Potential of the Brain Initiative. Good afternoon. I'm Katie Sale, Executive Director of the American Brain Coalition. I'll be the monitor for today's webinar. During this presentation, you'll learn about the Brain Initiative and new tools and technology being developed to study the brain and their transformative potential. These tools and methods are helping researchers expand their basic understanding of the highly complex and intricate brain, and forming a foundation for future progress on a wide range of neurological and psychiatric diseases and disorders that affect 1 billion people worldwide. This webinar will focus on one such tool, and presenters will discuss how, with further study, it could one day be used to advance research on developmental disorders such as autism. While we will be using autism as a vehicle to present the connection between the basic research, patient treatments, and patient advocacy during this webinar, there are a wide range of areas where the Brain Initiative is making inroads, including Alzheimer's disease and traumatic brain injury, to name others. Now I'd like to introduce our speakers. First, we have Dr. Spencer Smith, an assistant professor in the Cell Biology and Physiology Department at the University of North Carolina School of Medicine. His lab explores how neurons and the networks they form compute. Dr. Nancy Minshew is our second speaker and is a child neurologist and professor of psychiatry and neurology at the University of Pittsburgh. For the past 30 years, her clinical research has focused on revealing the cognitive and brain basis of the behaviorally defined syndrome of autism spectrum disorder. Finally, we'll hear from Dr. Robert Ring, Chief Science Officer for Autism Speaks, a leading science and advocacy foundation focused on causes, prevention, and treatment of autism spectrum disorder. Dr. Ring has specifically overseen the launch of numerous initiatives that have dramatically expanded the strategic scope and increased the impact of the science investments at Autism Speaks. To start, I'm going to pass it over to Dr. Smith.
Hi, thank you. I'm happy to share my research with you all today. A key motivation for the Brain Initiative is to better understand brain function at the neuronal level, and this basic research can provide new leads for treating complex diseases like autism and schizophrenia. In a moment, you're going to hear from Nancy Minshew about some exciting progress in clinical research for autism spectrum disorder. We all want to sustain and accelerate this progress. One way to generate new potential therapies for disease is to better understand the underlying hardware, the brain, and what goes wrong with brain circuitry at the neuronal level in complex diseases. This approach is complicated by the fact that there are still large gaps in our understanding of brain function at the neuronal level. Principally, no one knows how groups of neurons, distributed across the brain, function in concert so that we can perceive the world around us, make decisions, and behave in healthy ways. This gap in knowledge is due to technical limitations, and so a major priority of the Brain Initiative at this point is the development of new tools for basic neuroscientists. This is where my lab comes in. My lab recently invented the first microscope for flexibly imaging neural activity in multiple areas of the brain at the same time. This innovation is important because it lets us explore how brain areas work together, and I'm very excited to share it with you today. The key thing we achieved is a broader field of view. We can now see neuronal activity over a larger portion of the brain than ever before. The various areas of the brain work together. Their dense interconnections support conversations between brain areas. If we study only one brain area at a time, then we only hear one side of this conversation. And we all know how difficult it can be to understand what a telephone conversation is about if we only hear one side of it. The same goes for brain networks. We need to hear both sides of the conversation, and with the expanded field of view our new technology provides, we now can. What you see here is a video of neural activity in the brain of a living mouse. Each one of these blobs is a neuron. When a neuron is active, this is called spiking, it lights up. We've genetically engineered the mice so that their neurons light up when active like this. This imaging method is called two-photon imaging. This data is fascinating because we are observing a working brain as it processes information in real time, and we can see when each individual neuron is active. The resolution, being able to see each neuron, is critical. Each neuron in the brain exhibits a remarkable degree of autonomy. It makes specific connections to other neurons, and most neurons in our brains have been with us our entire lives. Complex dynamics play out across intricate networks that span the entire brain and underlie our thoughts and behaviors. Two-photon imaging gives us the resolution to see this in action. This neuron-level resolution, this level of detail, helps us develop accurate mathematical models to better understand how the brain works and tell us what might go wrong in complex neurological diseases like autism and schizophrenia. Now, you can buy an imaging system on the market today that will give you a video like this, but you can't zoom out. All you get is this peephole view. If you try to zoom out using a lower magnification, the neurons blur together and you can no longer tell what each individual neuron is doing. And let me give you a sense of scale for this peephole view. The width of this field of view is about 1/4 of a millimeter, or roughly the width of a few hairs. So this is a very small portion of the brain, even for a mouse. Before we developed our systems, scientists could only look at neural activity through a peephole like this. We wanted to break free of this peephole view and zoom out for a panoramic view. We'd like to see a large expanse of the brain all at the same time, so that when two brain areas work together, we can listen in on both sides of the conversation. For example, my lab is currently working on understanding how the mouse brain processes vision. And many of the important brain areas for vision are here close together, the lighter colored areas shown on the right, but still outside of this peephole view. When I set out to address this topic, the technology did not exist to zoom out and still deliver high-resolution neural activity. I figured out what was limiting the current technology and devised a plan for developing it, and in my lab, we invented it. I and my team have degrees in physics, mathematics, optics, and engineering, as well as neuroscience, and this combination of expertise lets us rapidly develop novel and powerful instrumentation for neuroscience, and this kind of interdisciplinary work for developing new tools is a key priority for the BRAIN Initiative. So, to build this new microscope, we had to start from scratch. We couldn't modify an existing microscope because they are so far from what we need. Microscopes are designed to image very small objects, and this is where they excel. However, their field of view is normally a lower priority. Many times that biologists are using microscopes, they're examining specimens that are dead or otherwise not changing very quickly. So it is often acceptable to take a single picture and then move the specimen and take another picture. But we can't do this when we image the brain. Patterns of brain activity change moment to moment, so we need to leave the brain in one place and use a microscope with a panoramic view. So to make this instrument, we went back to the drawing board and started from basic principles. Through extensive optical engineering, computer-aided optimization, custom fabrication, and high-speed electronics, we arrived at a design and built it. We call it the Treppin 2P, which is short for Twin Region Panoramic Two-Photon. Here's what it looks like in our lab. This is a front view on the left and a back view on the right. Let me tell you, this system worked better than I expected. We increased the field of view 100-fold. This is quite literally like going from a 1 megapixel camera to a 100 megapixel camera. The difference between TV in the 1980s and the latest super high-def 4K TVs is about 25-fold, and we obtained a 100-fold increase. I want to show you one of the first movies we took with this new system. But first, let me remind you what the state-of-the-art used to be. This is the field of view compared to 1/2 of the mouse's brain. This is the peephole view we were limited to. Now, see what we can do with this new system. So this is some of the first data we obtained. On the right, the field of view will be indicated, and you can see how many brain areas are within the field of view. And on the left is a portion of the video that is being zoomed in and out so that you can see that this system resolves individual neurons. Instead of just seeing one portion of one brain area, we can now see a lot more. Not just an entire brain area, not even a couple of brain areas, but nearly all of the cortical brain areas that are important for vision. And importantly, we can resolve individual neurons in each one of these areas. This is like Google Maps. We can zoom in and out, and the resolution we need is there. The black regions are blood vessels. We can't see through those, at least not yet. I wish I could share with you the sheer exhilaration we felt in the lab the day we got this data. It was like taking blinders off for the first time in my life. Suddenly there's a flood of new experiments we can now perform. We can truly examine how vision develops in the brain moment to moment through conversations between different brain areas. Let me give you an idea of what we're exploring at the moment. In my lab, we're focused on the parts of the brain that process vision. Mammalian brains, from humans all the way down to the humble lab mouse, process visual input faster and more efficiently than any man-made device. If we can uncover the computational principles involved in neural circuits for visions, then we can gain insights into how other parts of the brain work, and we can also examine what aspects of neural circuitry are different in autism and schizophrenia. When you see things, those images are analyzed by several different brain areas in parallel. We can now simultaneously image neurons in different regions and estimate what information they are exchanging. In our preliminary experiments, we have measured, for the first time, how these conversations between brain areas change depending on what the animal is looking at. There are some times that neurons in two particular areas exchange a lot of information, and other times that they exchange less. In summary, we have created a new microscope for analyzing brain function, called the Trepan 2P. With it, we can now zoom in and out and view brain function at different scales, increasing our ability to view brain activity by 100-fold. The Trepan 2P enables us to better understand the complex patterns and conversations among neurons in the brain. We are currently using the system to examine how different parts of the brain work together to support vision. We are also examining activity in developing brain circuitry to identify aspects of neural circuitry that are different when genes linked to autism and schizophrenia are changed in mice. Thank you very much.
Thank you, Dr. Smith, for highlighting the fascinating and important work being done in your lab. We will now turn it over to Dr. Minshew.
Good afternoon. My job today is to talk a little bit about what this basic research means to clinicians who are working with families and with individuals with ASD. Now, as most of you know, autism spectrum disorder is the term we now use for this group of disorders that result from underdevelopment of the skills for being social, communicating, recognizing and regulating emotions, understanding concepts, and functioning in a dynamic world. Now, current prevalence estimates are 1.5% to 3% of people have autism spectrum disorder. I don't think 3% is an overestimate at all because our current estimates do not include people who have a clinically significant fragment of ASD. In addition, though, the more that I have learned about these skills, the more I look around and I see that all of us at different moments or times have problems with these skills for various reasons. So we can learn a tremendous amount about social competence, communication competence, problem solving, and function in daily life by studying autism. Of course, our primary incentive and motivation is to improve clinical care and the quality of life for those with ASD and those who have skill deficits in these areas. Now, the first logical question one might ask after hearing Dr. Smith's presentation is, what does the imaging and mouse genetics work have to do with helping an affected family member? I'd point out that this is a question that has been asked about all research. And now, of course, we take for granted that research that was done in the past that changes the quality of care, of course we would do that. But at the time, that was not the thought. Now, I'd also say that on that same vein is that by definition, current research is about what we don't know and need to know in order to improve care in life. The benefits do not accrue immediately. They accrue as the research has accomplished its goals. It does take time. Now, we can, though, think about how has past research changed the face of clinical practice in ASD. And I'd have to say again and again, it's been a phenomenal change in what the clinician can say to families, how they can answer the questions, and what they can do. Because of past research, clinicians can now answer questions about prevalence, diagnostic criteria, methods for applying these criteria, a really great breadth to the syndrome that includes not just what is considered the core symptoms, but all the associated and comorbidity manifestations, how people with autism think, which underlies how they behave the way they do that is different from other people, that this arises from alterations in cortical systems connectivity. We can talk about hundreds of contributing genes. We can talk about manifestations in infancy, onset of brain changes in development during pregnancy. We can talk about effective treatments for most people, and the list could go on and on. However, what I would like to say is that It was a dramatic impact when we knew enough about how people with autism were thinking, how they perceived the world, and relate that to their behavior. We could then answer parents' questions about, why did my child do that, and how do we get him to act differently? We have answers to those questions, and in fact, we have interventions that are effective for children and adults. But I'd like to give you two other specific examples of how imaging and mouse genetics research has helped us now. First of all, we know that alterations in the connections of neural systems underlie all the manifestations of autism. But we also know, because of imaging and genetic research, that these systems are plastic or changeable in autism in both children and adults in response to treatment. We also found out from mouse models of autism that autism can be corrected if we understand the mechanism causing the autism. Now, what we know, though it is so much more than we knew 30 years ago, it's just not enough. And it's so not enough. And if you want to know how much more we need to know, just ask a person who has a family member or a neighbor with an affected person. Now, if we're going to take the next big step forward to improve lives of those with ASD and everyone else that has temporary or long-standing impairments in these skills, then we really need to understand the mechanisms in detail of the cognitive and behavioral basis of social competence, communication, and problem solving in the real world, the mind and brain basis of these abilities, how the brain develops these capabilities. We also know that the brain development is under close genetic guidance, and we need to understand those mechanisms in detail, and we need to understand this at a cellular and molecular level. Once we understand that, we'll have tremendous power to develop new interventions that will help a lot of people. So we come back to what we need to know from basic research is, what is the cellular and molecular biology of autism? This brings us face to face with complexity. The brain and DNA are so incredibly complex. It is complexity on a scale that's nearly beyond our comprehension and certainly beyond our daily experience of life. In some ways, all of us sort of know this. We drive cars, we use phones and computers. We take them for granted as functional entities. If we need a phone, we go to the store and we buy one. Only if they break do we begin to think about the depth of knowledge and skill over generations that went into creating them. So we do know that there is tremendous complexity in the world, but the complexity of the brain and of DNA trumps all that, goes so far beyond that. From my perspective, the brain is the most complex entity on the planet, and we need to understand how it was built molecule by molecule. Second thought is to remember that the program for brain development is all contained in that single cell that is the origin of all babies. How complex could that be? We need to know exactly how the brain functions, how it's built. Now, all this is knowable. We just look at the history of neuroscience. A lot of people working on a lot of different issues over time, and we've come to know a lot. But I'd say we need to expect the research that is going on now and in the future to be very complex and therefore seem obscure perhaps to us who were sitting in the clinician's chair talking to families. We know that it will take a lot of time and a lot of work, but in the not too distant future, we will have radically improved the treatment for people with challenges. Thank you.
Dr. Minshew, thank you for highlighting the very important role between the clinician and their work with patients. Excellent. We'll now hear from Dr. Ring.
Thanks, Katie, and good afternoon, everyone. Good morning to those of you who may still be in other time zones. Just wanted to start off by thanking the American Brain Coalition and the Society for Neuroscience for having put on such a wonderful webinar today. And you guys are certainly being treated to some of the finest work being done out there in the autism part of this larger brain initiative, hearing my colleagues Nancy and Spencer here. So what I'd like to do is offer, take a step up and offer a perspective from the patient advocacy groups. Really, if you think back to the president's initial announcements around this initiative, he called for this to be an all-hands-on-deck kind of response to that complexity Nancy was just mentioning around the brain. And really, like any successful research endeavor so grand in scale as the one we're talking about today, it really doesn't work or draw meaning without close partnership with those individuals that ultimately the science is intended to serve, whether or not it's just purely understanding the journey they are on with autism or wrestling with some of the challenges and opportunities that it brings. I work for a large patient advocacy organization, which also has core to its mission the support of science, and we see a real integration between that. And I think the success of the BRAIN initiative itself is really going to rely on the ability to form partnerships across this complex research ecosystem of different skills, different partners, etc. And really, we're all share in common this desire to want to return innovations and discoveries that are being made in labs around the world, insights and understanding around the brain into things that are going to have real-world application for individuals living on the spectrum, if we're talking about autism, or in the larger field of mental health and neurology. These real-world applications are medicines, they're new ways of diagnosing disease, new ways of treating and managing it. But really, to ensure that the technology development and the focus of research offer that promise of delivering on those, there has to be a voice of the end game, if you will, the patients in helping to shape the strategies and the priorities for that. And that's where we as patient organizations come in. We play a very strong role in helping to broker that conversation between the needs of patients and the larger research community. And that's really essentially what the BRAIN Initiative is trying to do. It's trying to bridge that world of discovery with that world of delivery that we're so dependent on. And it's, I think, why patient organizations are so excited about the BRAIN Initiative. And it really is part of this larger, revolution going on in front of us, whether or not you acknowledge it happening in front of you or not. The brain is at the forefront of the advancements in technology that are going to shape medical research for years to come, and we're getting a taste of that just today. What I thought I'd do with the time I had is just point to a few autism-specific things that are going on where patient organizations have their fingerprints, if you will, that are helping to drive forward the initiative itself. Patient organizations are a lot more than just working in Washington to advocate, on behalf of individuals and their families. That we, Autism Speaks, and so many other foundations are actually out there funding research. We're establishing and even directing collaborations. Some are even running their own research operations in-house. One area that I'd like to focus on is incredibly important, something that patient organizations do really well, is that's starting and maintaining resources, databases, and repositories that provide the reagents and resources that researchers depend on out there. But for a later time, if we have, there's a lot of really interesting work being done by patient organizations to serve this initiative, if you will, by supporting entrepreneurship and small companies that are actually trying to turn this into real applications. And we as advocates are channeling that voice to larger companies and helping them put autism on the map. A couple things I'd like to point out. One, the initiative around the brain, it struggles somewhat in that we're not like cancer where we can biopsy tissue and study it as easily as a disease that affects a peripheral organ. But if you're going to study a disorder of brain development like autism, you really have to have access to the brain. And although there's some very innovative ways that you can do that using animal models like we've heard today, brain tissue is an incredibly important resource for this entire initiative, not just for autism, but any other disorder. And we've partnered with the Simons Foundation to help build an international brain banking network that's providing that for the field. And we're also playing our role as an advocate in breaking down barriers to donation there. That's a very important role for foundations. Just to build off of what Nancy had said, I think the importance of the genome and understanding the risk for autism can't be understated. To help provide the field with sort of an unprecedented resource, we've actually partnered with Google to whole genome sequence 10,000 individuals with autism and their complete families. This is determining every letter of code for each of those individuals, which produces a massive data set. In fact, 10,000 genomes is equivalent to watching a high-definition TV show streaming continuously for 13 and a half years. It's massive, and that's why we've partnered with Google to make that possible. If I was to offer one sort of general theme to those of you on the line who are not as familiar with the technologies that we're talking about today, one common theme that dominates everything that's going on out there is we're in a world where the technology is creating data at a scale simply unprecedented, something we've never seen before. And most of the big biology problems that we'll face in the future are going to be problems that are dealt with by engineers and data scientists and are going to be made possible through informatics and technologies that help to maintain information. It's really incredible. I invite you to come visit our website, mss.ng, and learn more about this. These kinds of resources you will see more and more of as the story of the Brain Initiative moves forward. Can't understate how important the rise of wearable biosensors are playing a role even in this Brain Initiative. And I think as you watch stories unfold, you'll see an increasing use of technology in everyday lives where individuals are working beyond the lab to provide data to research studies, whether or not you're wearing sensors that monitor the peripheral physiological expression of stress and other brain disorders, or whether or not you're actually studying the brain itself in real time in a real world setting. These are very real aspects of what are going on in the field, and they're worth taking a closer look at. I'll just wrap up by offering an extension off of where Nancy brought us. As we think about treatment in autism, we certainly assume that treatment will continue to be multimodal. And what we mean by that is efficacy in managing symptoms of autism, which are very different across individuals in terms of their complexity and their intensity, are being managed effectively with behavioral interventions. They're being managed through occupational therapy and pharmacological treatments, drugs and medicines. But actually technology and the knowledge that's being created through initiatives like the Brain Initiative are really inspiring a whole generation of innovation that are taking us out of some of these traditional modalities of treating individuals. And one area that I think we should, I would encourage you to take a closer look at is this intersection between traditional cognitive neuroscience and the world of computer games. Some companies are actually out there developing game platforms that, much like you would be able to isolate a circuit in the brain visually using an imaging technique, or just like you would isolate a muscle group as you exercise a particular way, we're finding out that you can actually exercise specific parts of the brain that may be not working well using games and other kinds of exercises that are brain-driven rather than through muscle groups. And one company in particular, Achille, that our foundation has invested in, is actually developing a therapeutic game for treating cognitive dysfunction in autism. So this is a really interesting area, not just in terms of its therapeutic potential, but these games and games that you see online have the ability to collect information, data about an individual's performance, and may in time actually be used to help diagnose individuals or stratify the population into the subtypes of autism that we know to be out there. So it's a really exciting time, and I think you're really fortunate to be treated to some of the exciting advances that are going on. There is a big picture, and there is a whole ecosystem of partners working together from the lab to Washington on behalf of this. So it's an exciting time. It's a real pleasure to be here. I look forward to the questions we have after this.
Great. Thank you, Dr. Ring, and thank you for reminding us that at the end of the day, we're all doing this for the patients that we serve, which is the most important part. So, we've received a lot of questions, and I'm just going to dive right in and get started. Let's see here. Here's one question. I'd like to understand where we are in the discoveries now, but also what are our projections for the near future, and what should we expect?
This is Rob. I'm happy to take a first crack at that. Well, thanks to whoever asked that question. It's a really important one to be asking. And the thing I would say up front is this is a moment in time where we should be more hopeful than any other point in this story arc, if you will, of autism and science. We're at a point where the last 20 years of knowledge creation, if you will, that the autism field has experienced is arriving at a moment where we can really see how this knowledge can be reduced to practice. And this could be as simple as improved models of medical care for the complex condition that autism is, to the increase in clinical trials that we see among biotech and biopharma companies who are working to develop medicines for specific symptoms of autism. If you look at any of these areas of activity, they have grown enormously just in the past decade. And I think that's something that families should be very hopeful about. The key for us, just to tie it back to my presentation, one of the key goals for us in the patient advocacy world is just to make sure that the research community doesn't lose sight of that ball. That there is this very delicate balance between needing to drive, take risks and drive innovative research, which may take years to pay out, with the need to provide here and now solutions for families. And that balance, I think, is something we're now starting to get a good rhythm in across the autism field. So it's a very, very hopeful time.
Thank you, Rob. Okay, so the next question is, with the conditions affecting people, why do we need to study mice at this stage? Maybe
this is Spencer. I'm the one. Yeah, yep. I'll answer that. Although I thought sure, Nancy did an excellent job talking about it in her talk. Yeah, this is essential because we want to make rapid progress. If somebody comes into a clinic and is having trouble with memory, there are different hormone levels. There are different imaging modalities that a doctor can use to quickly diagnose and devise a treatment for that person. And it really required basic neuroscience and animals to understand how to develop those imaging technologies and how to develop those therapies. So this is the frontier. This is where things get figured out first, and then they are rapidly translated into humans.
This is Nancy, and I think I'd like to reiterate that as well. There are questions that we need answers to that we can't easily answer if we study humans, but we can if we study mice. And I remember some years ago there was a study of a mouse model of an autism gene, and the gene was introduced into several different strains of mice, and lo and behold, each of those mice had a different phenotype. And it brought home the influence of all the genetic background that is so important and influences how risk genes are expressed. And we wouldn't have been able to do that, or not so easily, if we tried to derive that from human studies.
Right, and I'd also like to point out that the Society for Neuroscience has an excellent website devoted to the importance of animals in research and can answer some of the questions there as well. So let me move right along. I am looking for specific recommendations for how to get kindergarten through college students interested in neuroscience and brain research.
I could start off with that. This
Sure.
is Spencer Smith speaking. In graduate school, I led Project Brainstorm, and one of the things that we did was an outreach program where graduate students would go around to area elementary schools all the way up to high schools. And one of the things that we would do is we had a short presentation where we would kind of talk about popular science and kind of accessible concepts about the brain. But one of the things that we did is we brought around preserved human brains. These were ones that had been donated by the patients and where we had approval to use them in educational outreach. And that was tremendously captivating for people of all ages to see a brain that somebody was a child with, grew up, fell in love, had a family, grew old, all with that one brain, and to see that in front of them and be able to touch it and understand that that's what made that person who they are really kind of makes this science tremendously immediate in a way that is difficult to do in a lot of other ways. So, for example, my background is in physics, and a lot of times in physics, I mean, my very far background in undergraduate, you're almost infinitely removed from what you're studying, these tiny little particles. And there's something so captivating about the brain and that being what makes us who we are that can really ignite people's imaginations and influence them to work on problems that they might not otherwise work on.
Thank you, Spencer. And I can piggyback on that, saying that my daughter's fourth grade class had human brains an exhibit, and it was fascinating to see the look on their faces, even at that young age, to be holding a human brain. It was really fascinating. So thank you. All right, moving right along. Here's another question from someone. What are some medical therapy options and alternative treatments for autism?
This is Nancy Minshew. That's rather a broad question, of course, for our short time frame. I'm not sure what is meant by medical therapy, but let me take a stab at it. It really depends on what the issue is that we're dealing with. We have some fundamental sort of cognitive behavioral interventions for infants and toddlers that are paramount. They're not widely disseminated yet, but the Early Start Denver model, for example, is a great example of our anchor treatment. For adults, we have cognitive rehabilitation sorts of programs. The Pierce program from UCLA is an example. Cognitive enhancement therapy that we're working on are examples. So those are sort of our anchors. And because we've demonstrated that these treatments do change the brain, I think we have to look at them as being as medical as other interventions. Now, in addition to that, of course, we have a third of people with ASD have epilepsy, so obviously we use anticonvulsants for those situations. If individuals have anxiety disorder or depression, we have medications for that. I think an exciting area of future medical treatment will be the direct brain stimulation methods, where stimulating the brain from the scalp painlessly will enhance the abilities that these people need and don't yet have. And so there's very interesting work being done on that. So there's an example of a very different medical treatment that is coming along.
Thank you, Nancy. We did receive a question with regards to the BRAIN Initiative. How can the BRAIN Initiative be leveraged to increase our understanding of developmental disorders and brain and behavior-related issues concerning children and youth?
Well, this is Rob. I think everyone here has just seen a wonderful sampling of research in different phases of that value chain, if you will, that brings discoveries ultimately to our community. And these are living, working examples of how our understanding of a developmental disorder like autism is being increased. And this is happening all over the place right now. And, you need only visit some of the Brain Initiative websites to see other examples of work going on specifically in the area of neurodevelopmental disorders. So it's a very sort of broad question also, but I think, here today we've seen a good example of some of that work.
Absolutely. And we're doing all that we can through the Society for Neuroscience and the BrainFacts site, the American Brain Coalition, to really leverage the Brain Initiative and the importance behind that. All right, moving right along. Dr. Minshew, here's a question for you. How can we make sure practicing physicians and advanced providers are reading brain research and talking about it with patients?
I think this question brings up another whole dimension to the issues. I mean, we have many clinicians out there in varying specialties, and brain disorders may be just one part of what they have to deal with. So we can't expect them to be expert in everything and to keep up with everything. And I think the challenge that Autism Speaks is addressing, and many other organizations, is how do we present what is a consensus that is translatable into guidelines to the broad array of people that need to know it in order to improve their treatment and do that efficiently. Because that's one of the true holes in our approaches currently is science has made a lot of progress, but the results of that have not been widely disseminated. And in part, it's because we don't have a source of funding that focuses on dissemination. So new treatments, no matter how effective they are, may only be available if you live within an hour of the center that developed it. So I think that begins to bring up, how do we effectively get guidelines to physicians?
This is Rob. If you may, I just would like to echo everything Nancy just said. I think the good news here is the medical community, through the efforts of things like the Autism Treatment Network, which is a program that Autism Speaks funds. It's a network of 14 centers across North America that are developing guidelines for the medical care of individuals with autism. Good news here is these guidelines are being developed now at a scale that we've not seen in years before. And not only are these guidelines being developed, but there's clear consensus arriving around areas of medical care that have been controversial for many years, and there's consensus forming around how we even measure the effectiveness of treatments, which has been missing for most of the history of this field. So it's a really exciting time. I might add to what Nancy has already said, in that you have to look at this in different directions. So in addition to the efforts to disseminate best practices and guidelines to practicing physicians, there needs to be just as much effort working directly with the community, empowering the community to be a voice for their own treatment and providing them with the tools and knowledge that helps educate their physicians. I remember just last year or the year before at the Interagency Autism Coordinating Committee, which helps to oversee the federal spending on autism research, I believe it was a parent suggested, with the issues of gastrointestinal complications in autism being very frequent, how do they get across, just basic treatments for constipation and bring greater awareness to physicians around that? Could there be just a card they could hand to a physician with a QR code that then took them to a website with all the known publications in that particular area to help provide the physician with the knowledge they're looking for? So there really are a number of very creative ways that we can be disseminating knowledge that is increasing around us all the time. Social media is another really exciting sort of platform for doing that I think the community will see us and other foundations working more in the future to get those best practices out. But it's good news in that these are being developed more than they ever have before.
Very good point about empowering the community. Dr. Smith, here's a question for you. Are there studies using brain imaging like fMRI to look at brain activity in children with ASD before and after different therapies or with and without therapies?
So I'm not a functional MRI researcher myself. I know that there is a lot of work in this area. What's happened even more is of studies of structural MRI, and here at UNC Chapel Hill there's actually quite a bit of work looking at brain structures, not activity, but the actual anatomy and how that changes in young children who are at risk of developing autism and either do or do not go on to later develop autism. And that has the potential to be a predictive tool and to start telling us what brain areas might be developing differently in children who go on to be diagnosed with autism. As far as fMRI, it's difficult to do that in young children. You need to hold the head still during imaging, and in babies a lot of times you can wait until they're asleep or at least quiet, and you can put headphones on them, but for other children, if you need them to respond to a stimulus, it can be difficult to perform those studies on a large enough cohort. But that is an active area, and I think there might be other people, maybe Nancy would be able to give a more contemporary update on that field.
Yes, I would add that there are studies that have looked at the brain pre- and post-treatment and have shown changes in connectivity and activation patterns. And that's part of what I was referring to when I said in children and adults, the new treatments are being assessed both before and after to see if it does change the brain. We want to know if treatments have just sort of superficially changed behavior or whether there's a real brain basis for the changes that we are seeing. And the answer is yes. In very young children, it's much harder, and researchers have resorted to electrical methods. I believe I saw one of the EEG electrode skull cap in Dr. Ring's slide set. So yes, that's been done, and there are studies in progress doing the very same thing.
Yeah, this is Rob. I'd like to add to this. I think it's a great question, only in that it invites, an extension of it. And what I mean by that is, this brain initiative, its very purpose is actually to expand beyond just fMRI and some of the agreeably innovative imaging approaches that we've had in our hands for many years now. Expand that toolkit of technologies available to study treatments, use it for diagnosis, you name it. And there are, I think autism is one of those areas where you're seeing it serve as a test bed for a lot of that evolving technology. And I think the Early Start Denver model that Nancy mentioned as an anchor for treatment has shown to impact EEG if you look through the lens of that modality. But there are other new technologies that are emerging that are going to be applied to this same question, and this is what makes this initiative so exciting.
Thank you. Nancy, we've got a question for you. Would you recommend any games for ASD kids, both using technology and outdoor activity?
I guess I would say I'd have a few suggestions, and it's more around what are we addressing. I think any game that promotes flexibility, do it this way now, next time do it a different way, watching other kids do the same thing so we build perspective taking, group problem solving where we encourage cooperation and begin appreciation for everybody has something to contribute and different kids will contribute. And then, of course, self-regulation, and I often think of the old red light, green light game or the Mother May I game that kids used to play long ago. I think those would work really well with children with autism. I think mindfulness meditation is addressing some of these issues as well, and I'm sure there's computer games that address these very issues. Rob may know more about that than I do.
but certainly none that have reached that level of evidence support that we all are looking for as a gold standard behind feeling comfortable making recommendations. But there are, Achille being a good example of a company, running an active clinical trial trying to build the evidence for that. I see the use of gaming platforms as a very, very exciting near-term horizon kind of opportunity for the community. It hasn't quite arrived there yet, but some really exciting things are right there on the horizon.
Great. Thank you. I think we have time for a couple more questions. Here's one. I'm a parent of a child with autism, and I just want to learn about the latest discoveries of the brain and how they may relate to his future or better understanding of his diagnosis.
This is Nancy Minshew. I'll take a first shot at this. What I recommend to families is that they go to the Autism Speaks website and they go to the Safari website, the Simons Foundation Autism Research Initiative website, where there are really nice reviews of all the emerging research. And the Autism Speaks website, of course, has many tools developed to help families with just this. The second thing I recommend is that hopefully a parent can go to some of the autism meetings that are held. Every state and even every city probably has these meetings, and they bring in speakers. And I think that's an excellent way to learn. And of course, learning from each other. When parents get together, they can talk about what things worked for their child and what things did not, remembering that every child's different. So something that didn't work for another child may work for your child. But that's a great way to find out about the resources in your area, is going to a parent group. So Rob, I'm sure you have other things to add.
Yeah, I couldn't have done it better myself in describing. I really, I think we've got some fantastic resources for families, toolkits, etc., that have been developed by clinicians like Nancy available. We do cover a lot of the science field on our website, so new discoveries being made all the time are covered. The Simons Foundation does a fantastic job on their website covering the science, field as a whole, and they do really well to break down some very complicated, topics in ways that families can take home and benefit from. Those are great places to start, and if you ever need help, feel free to reach out to myself. I might regret giving you that offer, but I'm more than happy to help connect families with information if I can.
Great. I'm sure some will take you up on that. We'll have one last question. How can brain initiative technologies help people with other diseases like cancer, Parkinson's, Alzheimer's?
This is Nancy. I'd like to take a stab at this because I recently heard a faculty member from UCSF talk about the dementias, and in particular she was talking about frontal temporal lobe dementia. And when she played a video of one of the adults, I thought, oh my gosh, I've seen people with autism do exactly what this man was doing. It happened to be he was counting ants in the backyard and kept careful tallies of the number of ants he saw on those scraps of paper. There were some subtle differences, but it reminded me that, the brain has a set of systems, a lot of them, but different diseases can impact the same systems. We see that overlap between ASD and schizophrenia, and we even see the overlap at the genetic level. So I think the Brain Initiative is aimed at developing our knowledge of these basic aspects of the brain, and they'll be applicable in different ways to all of these disorders.
Excellent. Thank you. Well, on behalf of the Society for Neuroscience and the American Brain Coalition, I'd like to thank you for participating in today's webinar. We're thrilled by the number of questions that we've received today. Please visit the BrainFacts.org site within the next few weeks for answers to questions that may not have been addressed. A very big thank you to our fascinating panel, Dr. Smith, Minshew, and Ring, for communicating the potential of the Brain Initiative. We hope that this is the first in a series of Brain Initiative webinars hosted on the BrainFacts.org site. Thank you again, and have a nice day.